Hello, welcome to the afternoon session of our conference on translational research on the brain. And I'd like to introduce Professor Peter Schofield. He's Executive Director and Chief Executive Office in Neuroscience Research at the University of New South Wales. And Peter was appointed the head of the institute in 2004. He graduated from the University of Sydney with the University Medal and was awarded a PhD in genetics from the Australian National University in 1985. He undertook postdoctoral positions in biotechnology in the US and in the University of Heidelberg. Peter was appointed an NHMRC Senior Research Fellow at the Garvan Institute in 1993, becoming head of the Neurobiology Research Program in 1999. His research interests focus on identifying genes that lead to mental illness and to dementia. In 1995, he awarded the Boehringer-Ingelheim Medal by the Australian Society for Biochemistry and Molecular Biology, and in 1997, the Goldschalt Medal by the Australian Academy of Science. And today he's going to talk about the genetics of bipolar disorder, discovery, prediction, and attitudes. Welcome.
Meili, thank you. Pleasure to be here. What I wanted to do this afternoon was to try to break my talk into two components. The first, without getting too bogged down in the details of the genetics, a little bit about some of the approaches we've been taking. And in a sense, I'm apologising in advance to tell you that the story is far from clear. We thought when we started we were going to be on a pathway which would give some greater some greater clarity and greater simplification about the understanding of the causes of bipolar disorder. We are learning things, but it's proving to be much more complex than we thought. That said, our ability and our interest in genes as individuals and as a community has grown enormously, and for conditions where they are highly heritable, we thought that we should actually start trying to understand a little bit about what real people, the community, understood and thought about that. So the second half, I'll talk a little bit about some of those results. So first off, bipolar disorder, also known as manic depressive illness. It's a common condition and it's a complex genetic disorder, typified by phases of both mania, and I think that the picture gives some greater illustration than any amount of words: euphoria, ability to do anything, but every so often comes completely crashing down, and then interspersed with periods of normality, periods of depression. And again, these illustrations out of the Cunningham Dax collection from Victoria, painted by individuals suffering from those conditions, I think really gives a sense as to both the euphoria of mania and the bleakness that depression can be. So about 1% of the population will suffer from bipolar in a lifetime. And bipolar is not a simple condition. It's teased apart clinically these days into four major groupings. Bipolar 1, which has both the profound manias and depressions. Individuals who have more psychotic symptoms, so schizoaffective disorder, affect being a disorder of mood. Bipolar 2, which has less pronounced manic periods, and unipolar, which is characterised by recurrent severe depressions. The condition, and again this picks up a little bit to Julio's comments earlier today, lithium, the mainstay treatment, was discovered by John Cade, but again it falls into that more than 50 years category. And so whilst some other mood stabilising drugs, valproate and carbamazepine, have come onto the marketplace. None were developed as mood stabilisers, none were developed for the treatment of bipolar. They were actually used originally as anti-epileptics and still are. And so we really are in this situation where, until we know a bit more about the condition, it's very hard to say what should the pathway of discovery be. And again, contrast that to some of the talks both before and after me with the understanding of the channelopathies in epilepsy, for example, or as no doubt we'll hear a little later, the role of amyloid in Alzheimer's. We do know that the condition has a major set of consequences socially, an increased rate of suicide, 15% higher than the normal rate, increased rates of comorbid conditions, be that substance abuse or other medical conditions such as anxiety, and then the personal and family cost in terms of breakups, unemployment, inability to hold a job and so forth. So 20-odd years ago, the old order Amish featured in this story because the genetic studies looked at large pedigrees, and the original study in the genetics of bipolar disorder was from a US group looking at the Amish and identified genetic linkage. In other words, a region on a chromosome where a disease gene ought to be located. Now, this perhaps shouldn't be surprising because if you go back to the beginning of last century, Emil Kraepelin, in a sense the father of modern psychiatry, who was responsible for separating bipolar disorder from schizophrenia as two clinical entities, and in a sense we still use that dichotomy that he created, said that he could see hereditary taint in about 80% of the cases that he observed in his clinic. So right back from the very beginning, one of the things that was most noticeable about bipolar disorder was that it had a family aggregation, and that's been shown over many years in twin studies, in adoption studies, by pedigree studies looking at the prevalence of, or the relative over-representation of the illness in large families, or looking at the population level. So I'm going to just very quickly summarise some of our most recent results. And in this particular set of genetic linkage studies, we've started with 35 reasonably large Australian families, 288 individuals in those families that have been recruited into the study, and of those, 130 have been affected with bipolar 1 or schizoaffective or bipolar 2, etc. So we've done the analysis both looking at the individual subclassifications of illness and also just aggregating them together. This approach of the whole genome scan was one that lent itself at that time particularly to pedigree studies, because you could look at the inheritance of genetic markers down the chromosomes on the one hand and the inheritance of the illness. And of course, if you've come up with a one-to-one correspondence of co-inheritance, then you've identified the causal event. And of course that approach has worked extraordinarily well for things like the Mendelian conditions. And again, Sam Berkovic's name has been mentioned today, and so many of his outstanding breakthroughs in the genetics of epilepsy have been because single large pedigrees or a small number of pedigrees have actually identified a single gene with a single mutation that is causal for the condition. And so that again was the rationale in which these studies were undertaken. As you can see, here on the slide, looking at the LOD scores, which is a measure of the odds ratio, the probability, in this particular cohort, and scanning across all of the chromosomes, the strongest signal was here on chromosome 15. It achieved just significance, and again, the sort of take-home message is most of these genetic linkage studies, whether they've been on pedigrees like this or done on smaller families, twin pairs or small nuclear families, SIB pairs, whatever, have all tended to come up with relatively small LOD scores. And immediately that starts to say, well, one of the reasons for this is that whilst this may be the location of a specific risk gene, the fact of the matter that the score is not phenomenally high is because there are likely to be many such risk genes. So one then needs to go in and map that region more precisely. And so here we are, we've zoomed in on chromosome 15, and we're looking at the cumulative LOD score in this multi-point analysis. So down here is no linkage, or a LOD score of 0. It's not no linkage, but there's no evidence of linkage. And you can see across this region of interest that the LOD score has climbed up above the sort of significance threshold here in the blue line, and there's a number of markers on the chromosome which have shown LOD scores up into the 4.5 region. So some reasonable evidence, and we sort of said, well, we should use the resources we have, and we should go and follow in on that region. So this region in genetic terms is 17 centimorgans. In physical terms is 6 megabases, 6 million base pairs. It's still a fairly large region to go and hunt through. But we did note that several other groups in the not too distant past had also found evidence of linkage to this region. The Genetics of Recurrent Early Onset Depression study had found almost an identical region. A UK study looking at a cohort of individuals with both bipolar disorder and with psychosis, perhaps better to say and or with psychosis, also had identified this region. And a couple of other studies using pedigrees that had either schizophrenia or bipolar or both, one from Italy, one from the US, had both also identified linkage regions near this general area. Whether this is the same region is perhaps up for debate, but this particular technique is not absolutely precise in its physical location. So let me try to untangle this picture or approach. Your DNA is a long linear strand, so in a pedigree, when you pass your DNA on to another generation, you have recombination that leads to breakpoints that creates the diversity that happens in meiosis. In a large pedigree, you can actually track from the preceding generations and down how that particular region of interest on a chromosome is being transmitted. And so the approach that we've taken here is one that you would also use in Mendelian genetics, of actually trying to follow that particular region of DNA and to map the haplotype or the ordered series of markers that are associated with the illness and the transmission at that locus in that family. So down this side of the diagram is each individual pedigree that was studied, and then along here we've got the different chromosomal markers along the bottom. So, for example, at the very top, in this family 10, the haplotype ran from up here to here, and then there was a recombination breakpoint, a meiotic breakpoint, that was then transmitted through that family, and it said the signal in this family is coming from that region. You then take the next family, so family 31, and the meiotic breakpoint is here, so the signal is coming from this region here. So if you take that approach in a Mendelian disorder, you can actually use that to narrow down very accurately and precisely, and usually quite quickly, the critical region. Bipolar and the other complex traits isn't a Mendelian disorder, so it's not quite as simple as that. But what you can do is use this to, in a sense, infer the region of greatest probability. So one family, and you do need to remember this is one family in the study, had a breakpoint here, another one had a breakpoint here. So it sort of, this approach gave us a larger region here, which essentially corresponded to a z-score -1, so a sort of reasonable confidence interval, and a smaller region here, which ended up being the high priority region. The bottom scale is not to actually megabase scale, it's just spread out. So when you put that into real space, it ends up, 6.2 region is here, and here are the known genes. And so here are the genes that are known in this region, and the high priority region from those recombination breakpoints is here with a relatively smaller number of genes located. So we then went and did, again, in a relatively small cohort, looked at SNP diversity, so single nucleotide polymorphisms in all of these genes spread across this region, so that we wanted to tag the SNP diversity in those genes. We also wanted to look at some of the intergenic regions between the genes, and we wanted to look at any other area of high conservation. So the results of that was all of the SNP signals down here, with a few in this area here coming out above the false discovery rate. Again, not a particularly high signal, but then we should also note it's not a particularly large cohort. So focusing in on those two genes of interest, this one here is sialyl transferase. So sialyl transferase is responsible for putting polysialic acid on carbohydrates, so your glycosylation on protein. And in particular, sialyl transferase has, as part of its function, putting polysialic acid on the neural cell adhesion molecule, so something that's very strongly implicated in cell-cell contact. The second gene down here is a solute transporter, and I have a less compelling story as to why it may be a gene involved in neural function. So I'll just leave it at it's a solute transporter. There is evidence from other studies looking both in schizophrenia, and remember some of those earlier linkage studies had shown the linkage signal was found in pedigrees either with schizophrenia or with bipolar. So in studies in schizophrenia in either the Japanese cohort or Chinese cohort had identified several SNPs in these regions that also showed significance, and so they've been added onto this figure. The problem being that the population differences meant that the SNP that was quite prevalent and seen in the Japanese population doesn't exist in the Caucasian population, so it's not actually possible to in that sense, map it. So evidence that the sialyl transferase gene may have a role from these studies, but just to put into context, the GenRed study, the Genetics of Recurrent Early Onset Depression, also mapped this similar region, different markers, but again I've put them on in blue, and you can see that their strongest markers came up in the solute transporter. So a range of studies providing evidence that this region, which is pretty much down to the two genes, may well be involved in conferring risk. In our cohort, the evidence all comes, or the evidence predominantly comes from the sialyl transferase, so we've put our initial efforts in there, and this work is really just starting now, so I don't have a completeness for it. But as I said, the role of sialyl transferase is to put polysialic acid onto carbohydrate attachments on proteins. It's been shown that sialyl transferase is decreased in its expression levels in hippocampus, and obviously this whole NCAM signalling is critically important in a whole range of areas, including neurogenesis, synaptogenesis, neuronal migration. So at least we're dealing with properties that you would start to think may underlie something like a major mental condition. Interestingly, especially perhaps given that schizophrenia has, in a sense, even stronger evidence for a developmental trajectory, and how early that developmental trajectory starts is unknown, but most schizophrenia presents itself in teens and early 20s. Bipolar disorder also presents itself typically late teens, 20s, sometimes into 30s. I mean, you know, again, that's the peak of when the presentation is. The developmental profile of the sialyl transferase gene is such that it is most high early in newborns, and over age its relative levels decrease. There is another sialyl transferase gene that then picks up at that point. So perhaps again, early neuronal patterning and that early expression, it may be that some of those genetic changes that are there in that gene are actually involved in perhaps causing that. So that's really the sort of state of play of that particular study, trying to tease out the individual pieces of variation in the group with bipolar versus the control group, and to then in cell culture type studies look at whether that impacts things such as migration. And then just a quick sort of jump forward into, well, what do we know about that cylyl transferase gene if you remove it in mice? And published work has shown that the mice do have behavioural phenotypes. And again, you would sort of expect that given the role of that particular gene. So I've got one more, I think, I'll just jump here. One more slide, which is really just to try to give you a very quick snapshot as to where the world is up to in this field. And a collaborative study using, instead of genome-wide linkage analysis, where you're looking at markers spread fairly broadly across and inheritance through families, the genome-wide association study, where you're looking at this point, hundreds of thousands of SNPs, and now, of course, millions of SNP markers, individual SNP markers, and looking for association across all the chromosomes against the disorder. And the highest profile candidate, the ankyrin gene involved with cytoplasmic anchoring and signalling, and the second candidate, calcium channel, were what were identified in a sense, the largest genome-wide study. There's just been a replication study of this, which we've contributed some of our bipolar families, and Nick Martin's contributed some of his control data to build up to this study, which has got a cohort size of 9,000 affecteds and a few more unaffecteds, and the ankyrin 3 gene is still coming up, but you know, this is one gene at a relatively modest p-value after it's been multiple testing corrected. This is hardly the genetic etiology of the condition. So perhaps another way of thinking about this is to say that you know, this might be the tip of the genetic iceberg, but in actual fact there are going to be many small genetic variants, each of which tip a little bit of risk or a little bit of protection, and when you start adding them all together, that may be a better way or more holistic way of actually trying to understand that genetic risk. Not very satisfying if you're trying to do a simple genetic test. The other issue in your genome is, of course, there are huge numbers of variations in terms of the way chromosomes are organised, and they're referred to as copy number variants, both duplications and deletions of portions of the chromosomes. And so in this review from Jonathan Sabat, based on his own work, in his sort of the illustrative, here's the deletion, a bit like I was showing you the haplotypes before, and when they've looked in to reasonably large cohorts for the presence or absence of deletions or insertions, they're relatively rarer in the control group, and they're relatively more frequent in certain locations in the disease group. And over here on the chromosomal ideogram, the dots are showing where there's a greater prevalence of insertions or deletions. But again, you can see that they're spread, a bit like linkage signals, across almost all of the chromosomes, often several places on a chromosome. And then when you get down to a single place on a chromosome, like this slide here, it's not a single deletion in the way that you know, the Philadelphia translocation gave such insight into leukemia, but rather a much broader range of genetic modification. So I think we've still got quite a journey to go. And so I just want to finish off on that section of saying, so we really are struggling with this problem of susceptibility genes. They're not disease genes, they're susceptibility genes. They're variants of normal genes. They could confer either risk or protection, and they're neither going to be necessary nor sufficient. And so it's likely that you will have many of these things. Of course, they could interact with other genes, they could interact with the environment, so gene-environment interaction. I mention that specifically because I'll move on to a second example of that in a moment. So, in a sense, finding susceptibility genes does seem to be a bit like this scientist here. It is an elusive problem. So I'm going to change gear slightly, and I'm going to deviate to a bit of science which took extreme public interest and in the end showed itself not to be replicated, after many, many groups had worked on it. But there's a portion, because of some of the other studies we did in a time window, where we were actually able to use this to start teasing out what people actually thought genes meant for their health and their mental health in particular. So the paper came out in 2003 from Caspi and colleagues, and it was based on the Dunedin cohort. It's a 1,000-person birth cohort followed in Dunedin, New Zealand. The individuals are in their 30s now. And the particular set of studies that came out from this cohort were looking at gene-environment interaction, so the influence of life stress on depression, and looking in particular at the serotonin transporter. It is a well-established fact that stressful life events are related to an increased incidence of depression. The more stressful events you have, the more likely you are to suffer from a depressive episode. The question is, are there genetic effects that overlay that? And this particular study said yes, there was. And as with a number of other groups around the world, we looked at how we might be able to replicate that particular observation. And in this particular study, it's from a relatively small number of school teachers, initially 150 school teachers followed in their, recruited in their first year of teachers' college, in the times past when you went to teachers' college after your sort of BA. And this group had been followed for 25 plus years. And so the genotypes that you have in the serotonin transporter were the S, the short, and the L, the long version, and of course it means you can have 3 different genotype combinations. And so looking at the proportion of the cohort that had been diagnosed with depression and the number of adverse life events that they had had, those with more adverse life events and the risk genotype in our study showed modest evidence of a gene-environment association. And this is an area of public interest. So Jonica Newby, one of the reporters on Catalyst, called me up sometime in between our study and the one from Caspi, and said, you know, this is really interesting. What I'd like to do is I'd like to get my serotonin transporter genotype, please, and I'd like to get my result, and I'd like to do that as the way that Catalyst does. If you watch that show regularly, you know that the journalist doesn't want to stand back and report on the story. They want to get right down deep into the story. So Jonica wanted to actually get her genotype live on the show. And of course, as always happens with journalists, she then said, oh, and by the way, can we do that next week? Love journalists. So we actually got the ethics committee to consider this on that accelerated time frame, and they said yes. And the argument that both we were happy with, as well as Jonica was happy with, was, in a sense, this is a bit, you know, if you're going to buy shares in a company, they've got to issue you a prospectus. If you've got a real lot of money, you don't actually have to issue a prospectus to those people, because the way the law works is it says they're knowledgeable people. That's how they got a real lot of money, and they can invest it however they like. And so basically we convinced the ethics committee that in that sense Jonica was both acting as an inverted commas knowledgeable person, and we did also put some backup support in there in case it was needed, and that this was actually an important way of getting the debate started. So Jonica did get her result live to air, and any of you that might have seen that episode would have seen that the result was palpable, and her face went the colour of the chair over here. And I don't mean the ones you're sitting on, I mean this red one here, because the genotype result was SS. Now, what did this actually mean? Because this is the great thing of a journalistic type story. It actually allowed the journalist to grapple with the thing, well, now I know, did I want to know, what will I do about it, and so on and so forth. But it was really, in that sense, she's like a sophisticated investor. It's not like, you or I might think about our superannuation fund. It's a very sophisticated person who has access to all sorts of scientific opinion and the backup of any counselling or assistance that was needed there. But it led us to this question, well, what would happen if we asked a real group of people? And we'd just done the teacher study by that point. So we actually gave them their results, again, with the consent of ethics. And we got this done before we gave them their results. So we asked them what their attitudes were. So they were one of two groups. They were either a receiver or a decliner. A receiver, I want to receive my result, or a decliner, I don't want my result. But this group of people had participated in this cohort study for 25 years. So just because they said they didn't want a result didn't mean they were going to walk away from the study. So they were still happy to say, I don't want my result, but I'm happy to fill in the surveys and have you come back in 3 months and 6 months' time and ask me what I thought about it and so forth. So the people that said no, they didn't want their genotype results. Sorry, no, I should just say, remember, these were school teachers. So this is perhaps not quite the normal community. Well, think about it in terms of, firstly, they'd all gone to university, so that's already removed whatever the percentage of the community is. They'd all done teaching. They were in their final year before they went out to school, so that meant none of them were in jail. They were all about to work. In fact, this cohort was actually started to investigate gender differences in depression. And they didn't actually find it, maybe because they'd become such a homogeneous group. No school teachers wouldn't say that, but relative to the whole population. So the decliners, the people who didn't want their result, said that they were more influenced by the fear of increased anxiety, negative effects on their mental health, their career. In other words, they didn't want the result because then they'd have to do something with it. It could be anxious. They might have to, again, we have a whole bunch of issues that come up with genetic privacy, insurance and the like. The receivers, in fact, had a little bit of a tendency to have higher neuroticism and anxiety scores. So, in fact, it was almost like, if I don't have it, then I'm going to be anxious and neurotic because I don't know what my result is. Because now you've told me I could have my result. Well, that's interesting. But the receivers were more likely to say yes to a test regardless of the gene effect. So we were actually able to ask them what they would like, irrespective of what the genetic outcome was, and then go through a process of counselling, giving them their test result and post-event counselling and follow-up. The other interesting thing was that most of the group overestimated their chance of being in the high-risk group. Everybody thought that they were going to have the risk gene. Of course, remember, at this point in time, this story was unfolding as very much this was quite going to be one of the best examples of sort of gene-environment interaction. But also, most people were quite pleased with their knowledge about knowing what their status was once they had it. I think I said this on this one before. Most overestimated their chance of being in the high-risk group. Those that, particularly in the high-risk group, who wanted to know what their status was, were pleased that they actually knew it. We also asked them, in the follow-ups, do you remember what your result was? Of course, that's a sort of pretty good way of deciding whether or not their attitudes match. And they actually did remember their results. And of course, as I said, school teachers may not be typical of the community, and certainly, this was a group of most of them were just sort of 40 to 50, like late 40s, early 50s. They were engaged in the study, had been for 25 years. That's not exactly your community of perhaps less engaged people. And then the coda to all of this, the meta-analysis of a whole host of these studies ended up concluding that and here's the interaction effect. So let me just work you through the whole thing. Here's the meta-analysis with the overall result down here, where one is no effect of the gene itself, and that was never under dispute. Here's the risk, the odds ratio, which is sort of like 1.5 of stressful life events from all of these particular studies. So stressful life events are a risk factor for depression. And then the gene by environment interaction, which over the meta-analysis fell back to 0. Here was our study, so you can see why we did this study. Now, as a collective, internationally, this hasn't held up. But as I said, we just happened to be in the right place at the right time to ask that set of questions in that particular cohort, and so we took that as an opportunity. And again, the world moves on, the world of linkage moved to GWAS, and so, you know, you only need to go on the web to 23andMe and do your little saliva tube and send it off in the post, and they'll mail you back a beautiful envelope and tell you what you're at risk for and not at risk for. And so there's this whole advent of sort of personalised genomics. I don't mean personalised medicine where the medical treatment is targeted against your gene type, but personalised genetic information, which is relatively unregulated and relatively unsophisticated in terms of what it says your risks or otherwise are. Working in the psychiatric field, one of the groups in the US set up a company to actually do this around genetic markers for psychiatric disorders, bipolar schizophrenia, and that created quite a furor in the community, and this particular editorial was in Science, or this News and Views article. So again, a fair bit of interest in the area, and as all good scientists, we had people proposing to do the tests and people saying that clinically responsible genetic testing in neuropsychiatry are bridged too far too soon. And again, everyone had really strong-held opinions on a zero fact base. So we sort of thought, well, maybe there's an opportunity of actually coming back in here and doing a bit more. And this really became possible because Alex Wild started a PhD with Phil Mitchell, myself and Bettina Meisser, and Alex had a background in journalism, having worked at ABC, so was really interested in this area. So the data collection's complete, and just waiting on some editor to say I'll publish it. So this was a large community study of asking people about their interest in predictive genetic testing. And we used the example again at that point of susceptibility for major depression. So the idea was to try to actually understand what the predictors and interests in having tests were in a random national population. And we asked in particular, and again, we used the serotonin transporter as the example in this study, and I think today that might be a little bit harder, although I'm not sure that the general public is that aware of the intricacies of the debate in this field. We asked them how they'd like to get the information. We asked something about their history and knowledge of mental illness, and we asked particularly issues about social stigma that might be associated with the condition. So it was a 1,000-person random telephone survey with structured interviews to actually elicit the information. And obviously these sorts of things come with demographics and clinical and family history. We asked questions about their causal attributions for mental illness, what they thought made it happen, what they thought that a genetic component would mean, and what they thought would be advantages or disadvantages. And I'm not going to show you all of it, but just 3 slides. So interest in how you got your information. So if we naive interest meant at the beginning of this part of the phone interview, we said, if you were going to get this genetic test information, where would you like to get it? Would you like to get it from your doctor, 60%, or would you like to get it direct to consumer, which is basically the internet? And 49% said yes. And then there was a standard bit of script that said, well, if you got it from your doctor, and you had a negative result or something like that, you could be referred to a specialist or a counsellor, you could be referred to a geneticist or a genetic counsellor, etc., etc. If you got it direct over the internet, well, you've got the report that you got given. And so then later in the interview, people were re-asked this question, and you can see a very small increase in the doctor, but a significant decrease in the internet. So again, this is trying to understand a little bit about how people might access this information, what they might want to do with it. We asked them about the perceived benefits. If the genetic test showed you were at lower risk than average for depression, what would that mean? One of the highest scoring was you'd get peace of mind. Always nice knowing that you're in low-risk group. At a relatively lower level, people said they would see themselves as being resilient to depression, irrespective of the stresses in life. If it showed that you were at higher risk, then you'd be ready to get early psychological help. You might be able to start minimising stress factors in your life. It might be able to tell you what treatment might be more effective for you, and it might also help legitimise depression as a biological disorder. And then we asked the perceived disadvantages, and here just on the higher risk group, and sort of going down in the percentages. Be afraid of being discriminated against by insurance companies or employers. You'd start worrying about depression that may never develop. I'd get depression. You may be more likely to feel depressed. That you wouldn't worry. You would worry that the result may not stay private. That you'd be worried about labelling or stigmatisation, and in fact, just knowing that you had a higher risk might in fact be a stressor that triggered depression. So when we did put all of that together, we came up with 5 factors that really described the best way of predicting the community interest. And so the 5 were: if you had a personal history of mental illness, you or someone close to you had suffered from mental illness, and so you had some greater awareness of it, you are more likely to want to know more about it. That the self-estimation of risk for depression, if you thought that your risk of developing depression was higher than what you perceive to be the average, you're more likely to get it. The endorsement of the perceived benefits of getting the genotype, if you thought that that would be useful because you knew something worthwhile about your genetic status or about your potential risk, then that attitude meant that you'd be more likely to want to actually get such a test. The belief in genetic evidence for mental illness would increase social stigma. So it's a little parenthetic that if people believed that evidence for mental illness will increase social stigma, they were more interested in being tested. Because you would have said that would have meant them being less interested in being tested, but unusually that was the case. And if there'd been a history of childhood abuse, so again, a major life stress event, but one that was actually able to be teased out more overtly. So quick sort of summary: strong public demand for predictive genetic testing for complex traits such as major depression. Same would apply for schizophrenia, bipolar presumably, despite beliefs about stigma. Demand for predictive testing for susceptibility to a mental illness is likely to come from individuals already aware of their risk. And the perceived benefits for predictive genotyping for psychiatric disorders appear to outweigh the perceived risks from those individuals. Of course, as the Huntington's disease example shows, even when you can offer testing, and of course in Huntington's there's no treatment, the uptake rates are much lower than what people say they will do, is much more common than what they actually do. And so again, in terms of the theme of today's presentation being about translation, this is another aspect about that translational journey. I think all of us working in the genetics of mental illness have been incredibly frustrated, but perhaps one upside of it is that it's actually increased community literacy about these things, as these particular studies have shown, and just being more literate about a condition is probably half of the battle, half the ability to actually go and seek treatment. So I just want to thank a few people. I mentioned Alex Wild, PhD student, who's been working on the genetic attitudes work with Phil and Bettina. Kay Wilhelm, who was the lead person behind the teachers study and the genetic attitudes there, and the genetic work, PhD student Erica McCauley with the silyl transferase gene, and a senior postdoc, Jan Fullerton, in particular. Thank you very much.
Thank you, Peter, for an excellent talk in genetic susceptibility in psychiatric disorders. Some questions? Sure.
I have a question, Peter. Thank you for the wonderful presentation. One thing that I'm kind of perplexed at with the current state of technology is that, as you showed very well with your pedigrees, the signals vary from family to family. So I think it's almost impossible for anybody that disagrees that all bipolar disorder is one single disease with one single genetic basis. So
There's actually a couple of parts in there. Firstly, in many conditions, Alzheimer's is a good example. There are rare familial cases that have presenilin mutations, for example, cause early onset Alzheimer's. Pathologically, the disease looks pretty much the same. Clinically, it looks the same. But you've got these individual families. There you have this additional feature of early onset presentation. We don't see that in bipolar and psychiatric conditions. So even, one way of tackling your problem is to do the whole linkage study in just a single large family. But even there, it hasn't come out that there is a single gene responsible. Completely agree. The more you aggregate everything together, the more heterogeneous the signal becomes. And so I think this is really going to come back to the clinicians and other ancillary markers that might allow subphenotyping clinically. So if you can have better clinical definitions, which at the moment, of course, aren't perfect, maybe you can use other measures, maybe imaging might be a scenario that you could add, a fairly expensive scenario. They might help you to tease that out. Our own view is that by taking pedigrees and only taking those pedigrees that show evidence of linkage to a region, we're trying to do that filtering that you're referring to maximise our chances. Not to look at the whole population, but to maximise our chances in those pedigrees. Yeah.
In your initial illustration, depression versus mania, going in plaque cell collection, in the manic one, you had did you notice there was a spiral in there? I'm thinking of Gino's with the sort of association with bipolar and migraine, particularly in women, as a key.
Yeah, look, I mean, again, as you point out, there's just those images are so laden with metaphor of the condition, both the mania one and the depression one.
There's a spiral in there.
Yeah.
It looked like a castellation.
Yeah. And I mean, that's down to the individual who's painted that particular one, and different images will have different things. But I just don't know the particular clinical story on that individual, but they may well fall into that category.
I'm just noting that there may be groups of people who have migraine bipolar who have to share it.
Yes, yep. And so again, there's this sort of issue that Julio raised of that may be the type of thing that allows you to tease out in the subsequent analyses. Probably better, Julio, still to have the whole GWAS data and then to be able to redo the analyses to try to tease out some of those phenotypes, or to use some of the endophenotypes that have been purported. Still don't know that endophenotypes have been the simple way through the problem either.
Peter, could you say something about what drives the selection of candidate genes?
Well, in the past, I mean, candidate genes were down to the horse racing analogy. You decided which one you thought had best form and put your money on it. Today, with the work I presented looking at the sialyl transferase gene, whilst we need to look at we're down at a candidate gene completely, but we have tried to use the genetic inheritance within those families under study to narrow the candidate understudy to the one for which there's the most evidence. Likewise, in the genome-wide association studies, the goal is to actually spread across the entire genome with markers and to find the one that comes out with greater statistical evidence, and then, in a sense, to latch onto it as your candidate to provide independent validation and verification. So I think our word candidate gene has perhaps evolved a little bit over the last decade or more from being, I've picked a candidate because it's my personal favourite, to this candidate has arisen because of, hopefully, a relatively unbiased analysis of the data, and it is the strongest candidate, so I will pursue it first and foremost.
Are you able to say what's happening biochemically
Yeah. Look, I guess the short answer is no. And this has been, in a sense, one of the reasons why the genetic studies of bipolar disorder became relatively so attractive was pathologically, imaging studies, blood biochemistries, brain activities were all really without a compelling hypothesis. The most compelling piece of data was the heritability of the condition, which led to the genetic studies. However, now that we're, in a sense, out the back end of a fair bit of that genetic work, I sort of feel that we're almost back to this same conundrum again. We really don't have a really good, strong lead as to the etiology, the causality or the pathology of the condition. And so I think this is a challenge and a problem that we got ourselves into, but one that we need to sort of work through to actually tease something out.
The Nobel laureate, his name I should remember, who did the work on telomerase, she seemed to imply if you use stress as a sort of diathesis, that stress might lead to genetic instability. I'm wondering whether you have any thoughts about that in terms of, if you get a hunch on
No, I think it's
sections connecting with other sections.
Yeah. I'm probably not the person to give that lecture, but in a sense, that's the epigenetics lecture. The modification of DNA by environmental effects and the telomerase type story would fit into that. And again, that may be another way of trying to approach the problem of pulling out a particular risk factor that whenever you've got one good risk factor nailed down, finding the second one becomes proportionately easier because you've got one piece nailed down. I think one of the problems that this whole field has got caught in is that the approaches that people thought might nail down some of the risk factors have proved to be far more problematic. Listen to the amyloid talk. Wouldn't the psychiatric disorders love some amyloid? Not that people would love amyloid in their heads, but having one good factor that can allow you to pin something down gives you a lot of control over the rest of your studies.
All right, there are no more questions. I'd like to thank you, Peter, for your excellent presentation.